So during RNA processing, you're going to edit the mRNA, and you're going to edit both sides. You're going to edit the 3' end, and you're going to edit the 5' end as well in very specific ways. Each end is modified. The 5' end is going to receive a modified nucleotide on its end. It's called a modified guanine is added on. And on that 3' end, you're going to get a poly A tail. So that is our sequence of repeating adenines. So you literally just get like, I don't know, it's like 10 to 30, something like that, adenines that are added on to the 3' end. There are several functions of this. We're not completely sure about all of them. For one thing, if this doesn't happen, it's not going to get exported to the cytoplasm, so it's going to get stuck in the nucleus. And sometimes that happens, especially if you're at a point where that protein is no longer needed. You don't want to keep making more protein, so that editing will shut down and it won't be processed anymore. It won't be made anymore. The other thing that happens is it's the 5' end. The 5' end is going to help protect the mRNA from hydrolytic enzymes. What is hydrolytic? Hydrolysis. What is hydrolysis? Breaking something with water. So these are enzymes that are working with water to break apart molecules. And these are found in the cytoplasm, and they're there to attack or destroy anything that doesn't belong there. So having that cap on there tells the enzymes this particular molecule, this mRNA, is supposed to be here. We don't want to break it apart. We don't want to dissolve it. They also help the ribosomes attach to the mRNA. And that poly A tail, when it says it appears to help it facilitate it to get it to the cytoplasm, the poly A tail appears to work as kind of a code. You know, like you got a gate code and you got to enter in the gate code. Well, the gate code is really easy, A. It's like 1, 2, 3, 4, 5, 6. But it has to be there, and if it's not there, it won't let it leave. So those are some things that are going to happen. But there's one more thing that we need to talk about, and let me actually move to that slide first, and I'll come back to 35 in a second. Most eukaryotic genes, and this is important because this is in eukaryotes. This is not in prokaryotes, and that is a question that they like to test you on. They like to say prokaryotes, and then things change with regards to what your answer is going to be. We're talking about eukaryotes here. So most eukaryotes have areas within their gene that are called non-coding regions. And they're called introns. They're in between the coding regions. Introns are in between the coding regions. So when an mRNA primary transcript gets generated, it has these intron sections in it. And they need to be removed before the mRNA becomes what we call mature. Okay, so it's ready to leave the cytoplasm, in other words. So those introns have to be removed. The other areas that are coding regions are called exons. And the process of taking out the introns that are in between and putting the exons that will be expressed together is called RNA splicing. And that is accomplished by enzymes as well as some other molecules that I'm going to talk about in a second. I'm just going to go back up to this picture here that I didn't look at. So now we've got, this is an mRNA. Okay, so this is an mRNA. Let's write that down. mRNA. And here I have my modified guanine. Okay, that's on my 5' end. That's typically called the cap, the 5' cap. And then I've got this poly A tail that, they're saying 50 to 250. Wow, that's a lot. A lot of A's are added on. And then I have a couple of different colors in the middle. I have this little area at the very beginning, and it's hard to see, and obviously yours is not colored, but it's shown as green here because that is our start codon. What was the enzyme that was going to produce mRNA? Remember what it was? Polymerase. It's RNA polymerase. So RNA polymerase is the one that is going to start this whole process and make this strip. But it needs to know where to start. So there is a letter sequence that will tell it where to start, and that is the start codon. Okay. Whenever the polymerase sees that, it knows here's where I start. Okay. Then there's also a stop codon, and that is a sequence, and that one's a little less able to see that one because it's red, and it's red already. But that's where you're going to stop. The RNA polymerase says, okay, I'm done. My job is done. I'm out. And then there's these little areas that are kind of pinky here that are pretty sure those are the introns. I just want to make sure let me just make sure that's what they're using for colors. Yeah, they are. Okay. Yeah, so the pink areas are called introns, and you can see they're in between those red areas. And we need to take those out. So we're going to take those out, and that's shown here. And we took out this section here and this section here because we don't want that. These are non-coding areas. Now, I'll talk about why those introns are there in just a minute. But a couple more slides about why they or how they're cut out. In some cases, RNA splicing, so cutting out the introns, is accomplished by something called a spliceosome. Spliceosome, isn't that a fun word? Nope, that's not where I wanted to go. Oh boy, that really took me back. Why does it do that? I don't know. Doesn't really matter. All right. Spliceosome. Spliceosome is made up of a bunch of proteins, as well as there are these little pieces of RNA. Now remember, RNA is how many strands? One. So there are certain RNAs. When they first started discovering RNAs, they thought there was only 3 types of RNA, or 2 types of RNA, and then they started finding other RNA. And so this is one of these types of RNA. And this RNA actually starts to break apart, or to take out, I should say, the intron areas, or the parts that do not belong in this particular mRNA sequence. So there are also some of these RNAs are catalytic. What does that mean? They not only start it, but they speed it up. So they're catalysts. What did I tell you? What did I tell you enzymes normally are? What are they made of? Amino acids, which is which macromolecule? Proteins. So mostly, like, I don't know, 80% of the time, your enzymes are going to be made of protein. But this is a special example where it's not made of protein. It's actually made of RNA instead. And these things are called ribozymes because they function as enzymes would. So they are enzymes. But their job is to cut the RNA and to cut out those introns, those areas that we don't need. So this was a big thing back in the day because everyone thought that the only enzymes in the body that we had were proteins. So if it was a biological catalyst, an enzyme, it was a protein. And it turns out that no, there are these things that are made of RNA that serve as enzymes as well. Picture before that I skipped over here is an example of a piece of mRNA being spliced. And inside here we have, inside the spliceosome, whoa, that is not what I wanted to do. Inside of there, I have these little bubbles. I'm going to change colors. That are RNA, and they're going to bring this loop together. And when they bring that loop together, they're going to cut the sides of it. And they're going to take out that intron so that I end up with 2 exons and nothing in between. Okay, I just end up with a piece that's going to be translated by the ribosome. Okay, that's called RNA splicing. Next page or next slide you can remove. We don't need that. It's just talking a little bit more about ribozymes and how they function as enzymes. You can read over it. There's nothing wrong with it. It's just we don't need it. So the thing that then becomes the question that some people will have raised in the past is, well, why are there intron regions? This makes this process more complicated. If there weren't those introns there, we wouldn't have to worry about that. Well, there's a couple things. First of all, introns may, in some cases, regulate gene expression. So those regions could be made into proteins. And what they do is they actually then come back to the mRNA or to the DNA to either turn on, make it easier for that gene to be expressed, or to turn it off to make it harder for that gene to be expressed. And we'll actually talk about that probably into next week when we get to gene regulation. The other thing that is important is the fact that what is an intron in one type of cell can become an exon in another type of cell. Okay, so here's my analogy. You have we're gonna be using cookies for analogies in this case, and then I always get can we have cookies? So you have a cookie recipe. What is always in your cookie recipe? Flour, unless you're gluten free. But let's say you're not. Flour. What else? Eggs, almost always, yes. Sugar. You can't have a cookie without sugar, right?
Chocolate chips.
Oh. Anybody not like chocolate chips? My wife hates chocolate chips. She hates chocolate. She's crazy. She cray. Okay, so we need butter. Oh, sorry, I said that. Butter. We need butter. Butter for a good cookie. Flour. What did we say? Yeah, salt usually, or some baking powder. Yeah. Okay, so those are ingredients that no matter what kind of cookie we need, are gonna make, we need those, right? We need those. We need the flour. We need the sugar. We need the butter. We need the eggs, etc. But if you're Lucas, you want to have chocolate chips. Oh, you would. Okay. Anything else we could add?
I wouldn't suggest it.
Raisins? Well, okay. So let's say we're not. Are we going to have chocolate chip and raisin? No. Okay. So let's get rid of the chocolate chips. Sorry, Lucas. And we're going to put raisins in. But what else do we need in there? Oatmeal. Yes, we need oatmeal, right? Now we need oatmeal raisin cookies. Okay, there we go. Okay, strike that. We're not making chocolate chip. We're not making oatmeal raisins. What else can we make? A sugar cookie. Oh, we can make a sugar cookie. We'd want to up the vanilla in that one, right? A snickerdoodle. We'd want to add some cinnamon in there. Okay, so this is an analogy to what is happening in this mRNA. So you have a gene, and it's a stock gene that has, it's like a recipe that says you can make it this way, you can make it this way, you can make it this way. You can make snickerdoodle, we can make oatmeal raisin, and we can make chocolate chip. Okay? But depending on what we're making, we don't necessarily want all of those ingredients at the same time. Okay? That's where we come up with the introns and the exons. So in some cases, those exons are always there. We always need the flour. But in other cases, we're going to have chocolate chip, we're going to make that an exon. But in other cases, we might want to substitute that out, and we might want to put in oatmeal and raisin. Okay. in different cells in the body, proteins may need to be tailored differently so that they can function within that cell. So you may need to take out different pieces of that recipe. Different introns need to be taken out. Does that make sense? Sure, a little bit. That's called alternative RNA splicing. You go to your cupboard and you're like, yeah, I don't have chocolate chips. What else can I make? Okay. Proteins. Back to proteins again. Ugh. Proteins have domains. Domains are parts of the protein that will have distinct shapes, distinct structures to them. And oftentimes, each exon will code for a different part or different domain within the protein. So, different part of the structure. So, if I look here, wait, what? Domains are areas that have a particular three-dimensional shape. And you can change these up, right? So you can take out one exon, and that will take out one part of the shape, and it'll give you a different protein. Here's an example that I have a gene, some generic gene, and I have a couple of introns. Got one here, and I got one there. So I'm going to get rid of these guys. I'm not going to make them. But I've got one exon that will give me this particular domain. I've got a purple one that will give me a different one, and a third one that will give me this last orange shape. Okay. And that can be changed around, and maybe now it would be easier if I had 4 of these. But I could suddenly say, okay, well, let's take out the purple, and now I'm going to make this into my third exon, and that would give me a different shape, and that would give me a different protein. Okay? So it gives you a lot of flexibility with the same genetic sequence to give you a different protein depending on what you need. Yeah.
So if you were to swap them, the purple would become the intron.
Yes.
Would become the exon.
Yes. So then this would become the exon.
Okay.
Say X. It wouldn't be denatured, but it would give you a different protein for sure. Yeah, and the three-dimensional shape would be different. And the last line of the previous slide actually says that this can happen, this exon shuffling. And so if suddenly, for whatever reason, now you're making a different piece of that gene, you could end up with a different protein being produced, and that could cause it to not work. It wouldn't strictly speaking be denatured, but it wouldn't work. Or it could give it a different function that it didn't have before. And we do see that when we get to evolution. We'll see some really weird things that we think different genes have changed over time to the point where they're producing different proteins that give different outcomes from what they used to give. That's kind of cool. All right. Now I want to show this video on. You don't have this one here. Yeah, we can probably hear it through the. Glad you asked. messenger RNA. All right, I'm going to stop there because that's as far as we've gotten so far. Does that help? Kind of see it? Yeah, I can. Remind me when I'm done.
Wait, so they remove an intron and then replace? No, they don't replace.
They don't replace it. They're just going to put the exons that there's a gap in. They'll put them together. Yeah.
Where do the introns go? Away?
I don't know for sure, but I think they're recycled, so they're broken down, so those nucleotides can be reused for more mRNA transcripts. Okay, so we've done transcription, we've done mRNA processing or editing. Now we need to talk about translation. It's going to be accomplished in the cytoplasm, and it can only get accomplished with the help of another type of RNA that is also single-stranded. They're all single-stranded, but this one has kind of a funky shape. It's called tRNA. T stands for transfer. Because what it is doing is it's transferring an amino acid from the cytoplasm to the ribosome where it will attach to make this polypeptide that will be turned into a protein. Okay, so it's transferring an amino acid to the polypeptide chain. And it's a fairly complicated process. We're going to go through it step by step. And I don't think that picture is really helpful at this point because we haven't talked about anything. So let's just look at the structure first. So each tRNA molecule enables translation of a given mRNA codon into a certain amino acid. The tRNA carries a specific amino acid on one side of its physical structure. And on the other side of that physical structure, there is a anti-codon. Now, what did I say a codon was? Remember that? Yeah, so those are the three bases. I need three bases to end up with an amino acid. That's for mRNA. So mRNA only has codons. Okay? The tRNA is carrying the complementary nucleotides to that three-letter codon. And so if it's a C, it's carrying a G. Okay, and it's three letters because we know three letters are what you need for an amino acid. So they are called not the codon, but they're called the anticodon. That's the complementary copy to the mRNA, 3-letter sequence. It's not really terribly important for you to know what the tRNA looks like, other than you are going to be drawing them. So, and I think visually it helps to see that. These are 3 different drawings. You won't have to remember what each one of them are. I just do it more so you can see a visual representation. My personal favorite one, it does really look more like a. So if you just look at it as 2D, it looks kind of like a three-leaf clover. You got this guy here. If you do it more 3-dimensional, it looks like that. I personally like the sock. I think it looks like a sock. And that's just another representation. That's the symbol they use in the book. I think in terms of drawing, and like I said, you are going to have to draw it. I think this is the easiest one to draw. And then you're going to have on one side, this is kind of a little one, but we'll make it bigger here. There's an attachment site for an amino acid. Now, every tRNA has a specific anticodon that it is carrying. And maybe that anticodon is AAC. Okay, AAC. I can't tell that that's a C. AAC. And that tRNA is always AAC. That's its anticodon. So it has to find, what does it have to find? What codon is it going to match with? A is going to match with, not a T, because now we're not, we're matching with mRNA. So our mRNA is going to be what? U, G. Ugh. Okay. And it's always going to look for UG. Always going to look for UG. And it's always going to be carrying the amino acid, and I don't know what it is, that corresponds to UG. Okay, and we can actually find that. You want to find it? Let's find it. It'll take a second. Not long. There it is. Yeah, it doesn't always want to erase. Okay, so it's looking for U, first one, second one, and third one. It's looking for, oops, that's not it. Sorry, UG. That's Lu. Okay, did you see that? Understand that? First letter is U, second letter is U, so we know it's in here. And then G is the last one, so we go over. And it says L, so it's LEU. Okay? Yeah. All right, let me go back down to, where was I? Here. Sock puppet. Okay? So it's always going to be carrying Lu. It's always going to be carrying Lu, and it's going to be looking for UG in that particular case. So every tRNA is just kind of like stuck carrying the same amino acid. Okay. Okay. In order for an accurate translation to happen, you have to have 2 things that happen. First of all, there has to be a correct match. So that tRNA and the amino acid has to be right. Okay? That particular tRNA has to find a Lu, an LEU. If it doesn't find it, if it brings the wrong one, that's going to be a problem because it still has the same 3-letter anticodon. So it's going to be bringing the wrong amino acid to the party, and that's not good. That's going to be bad. Second, there has to be an accurate match between the tRNA and the mRNA. Okay, so UG has to match up with AAC, and if it doesn't, then that's going to be a problem too. There is something called wobble. And wobble is the idea that oftentimes that third codon is not as important as the first two. What does that mean? Well, let's look at that slide again, going back to that codon chart. This is, by the way, a codon chart. Call that a codon. Oh, not that. Codon, so you're going to read from here. You're always going to read the mRNA. You're not reading the tRNA. You're not reading the anticodon, so don't get confused by that. And now I forgot what I was talking about. What was I talking about? Wobble. Okay, yeah. So, for example, proline's a great one because yesterday I used proline, and it kind of burned me. Because if you look at proline's in the middle here. Which is the least important base in that one? The first or the second or the third? Is the third base really important? No, it needs to be there, right? If it's not there, then you get what's called a frame shift, and that's really bad. But as long as there is something there, it doesn't matter if it's U or C or A or G, it always codes for proline. So that is what they call wobble. And the reason that's important is because living organisms do have things get messed up. DNA does get replicated incorrectly. There are things in the environment that can cause mutations. And a U or a C might change to an A. And if a C changed to an A in the DNA, it wouldn't matter because you'd still produce proline. It would be what we call a silent mutation. We'll talk about mutations later. But it wouldn't make a difference. So that's really nice because it means that our DNA is resilient to errors because of that wobble effect. Same thing goes for glycine here. That last one, again, is not important. It still codes for the same thing. Arginine is the same way. Okay, so a lot of them, it doesn't matter what that third one is. It's still going to give you the same outcome. Okay, wobble, wobble, wobble. All right. I might stop there because we're getting to the point where, yeah, wobble. All right, yeah, I'm going to stop there because we're going to have to get into ribosome function and structure, and that's going to take a while. We're about done, so we're going to stop there.